Hi, today's topic of discussion is the general principles of solute and water transport across the epithelial cells of kidney. First, we should know how urine is formed. In this schematic diagram, here this is the Bowen's capsule, this is the glomerular capillaries. The formation of the urine involves three basic processes. First is the ultrafiltration, next is a reabsorption, then third is secretion. Ultimately, combination of these three forms the urine. If GFR of a person is 180 liter, which is completely protein free, out of this, less than 1% of filtered water and sodium chloride with the variable amounts of other solutes are excreted in the urine. By the process of this reabsorption and also secretion, the renal tubule actually modulate the volume and the composition of the urine. That is very important. So reabsorption and the secretion actually modulate the formation of the urine. Okay, so we have to know more about how the solutes and water are reabsorbed and secreted. Actually, by this process, the kidney control the volume, osmolality, composition, and also the pH of extracellular and intracellular fluid. Now the recapitulation of tubule, renal tubule, and pericapillaries and peritubular capillaries and peritubular vein. So this is the afferent arteriole, this is the efferent arteriole. After entering to the Bowen's capsule, giving the glomerular capillaries from where the filtration is happening. Then when it is leaving, this becomes efferent arterioles. This is winding up this tubule. Then it is converted to peritubular vein. Now you see here, this is a proximal convoluted tubule. This is the late part of the proximal tubule. The first part is convoluted one. The straight part is proximal tubule, but it is not convoluted. This is the descending limb of Henle. This is the thin ascending limb. This is the thick ascending limb. This is the distal convoluted part, and this is the collecting part, as we all know here. Now the reabsorption is happening from this, mostly from this convoluted part of the proximal tubule, the glucose, amino acid, sodium, and the water. Okay, now check it here. In this descending limb of thin part of the descending limb of Henle, what is happening? There is no solute, only water is reabsorbed from the tubule to peritubular capillary. And coming to this thick ascending limb of Henle, there is no water reabsorption, only the solutes are reabsorbed to the peritubular vein. Coming to here whatever it is happening, again few solutes are reabsorbed, but some are secreted to the tubule also. What are the main component of the secretions? Mainly the drugs, if the person is under any drug, and then it is hydrogen ion is another secreted solute, potassium also. And the later part is a collecting duct, the water and also sodium, it is also reabsorbed with the effect of ADH, that is vasopressin, and also the effect of aldosterone. The sodium is also reabsorbed from this collecting duct. We will discuss in detail in coming slides. Before going to the transport processes, we must know what are the mechanism of the tubular transport. This is a recapitulation of the general physiology. Again, we should know in detail. First is active transport. As we all know, the active transport is two types. First is primary active transport. And why it is called active transport? Because it requires energy and it transports the solute against the concentration gradient with the help of the energy, that is ATP, and pumped out of the cell, or brings the solute from outside to inside the cell. An example, most important or general example of this primary active transport is sodium potassium ATPase pump. Another one is secondary active transport. As we all know, why it is called secondary active transport? Because the primary active transport, with the help of that primary active transport, it produces the chemical gradient, and by utilizing that chemical gradient, secondarily some solutes come inside or goes outside of the cell, so it is called the secondary active transport. Then other process, the secondary active transport are mainly two types. One is a co-transport, or called symport, means two solutes are coming in the both same direction, or counter transport or antipode, opposite direction. Two solutes are moving with this chemical gradient, with the help of this chemical gradient in the opposite direction. That's why it is called antipode. Then other mechanical transport process are passive transport without requirement of any energy. Mainly two types. One is osmosis and another one is diffusion, and another one is a pinocytosis. We'll discuss what is pinocytosis. Passive transport. Passive transport, as I have discussed, the passive transport is without requirement of any energy, right? So actually the other way of telling this passive transport is diffusion. It's along the concentration gradient without requirement of any energy. Yes, there are some transport mechanism which is called the facilitated diffusion also, which requires some carrier, right? And the osmosis of the water, this is also passive without any requirement of energy. And passive reabsorption, example of another passive process is passive reabsorption of urea. Now the pinocytosis. Pinocytosis is an active process. It is, other way you can tell it as a cell drinking. So what happens, cell takes in the fluid along with some dissolved small molecules. In this process, the cell membrane folds and creates some small pockets and capture the cellular fluid or the dissolved substances like the protein and peptide. And actually this pinocytosis, it happens in proximal convoluted tubule. As I have discussed, the kidney is the organ which maintains the extracellular fluid volume. By how? That is mainly the sodium. And sodium is one of the most contributor for the maintenance of this extracellular osmolality and the volume. And we should know about the not only the sodium, other solute, the concentration of the other solutes in the tubule after filtrating through the glomerular membrane. So now the concerned topic is filtered load or tubular load. First, you should know that very tiny fraction of sodium is present in the urine. Okay, if we check the urine, normal constituents of the urine, is the very tiny portions of the sodium of extracellular fluid is present in the urine. It's coming out through the urine. Now, how it is happening? What is the, for that first we have to know what is the filtered load of a particular solute. The calculation of this filtered load or the tubular load is GFR multiplication of the plasma concentration of that particular solute. Now if we take for example the GFR is 180 liter per day, and the most common constituents of this filtrate is sodium. The concentration of the sodium in the filtration is completely the same in the plasma. It is 142 millimole per liter, because whole the sodium is filtrated through the membrane. Now the filtered load is, ultimately it is coming to approx 25,500 millimole per day. Just think it. It is actually, you will surprised to know that it is 1.5 kg of table salts. Isn't it very surprising? The tubular load of sodium at the beginning part of the proximal convoluted tubule is 1.5 kg of table salt. But compared to that, do you know the millimole concentration of the sodium in the urine is only 42 to 20 millimole per day, depending on the types of food the person is taking. So what is happening and how it is reabsorbed from the tubule to the blood, and what the mechanism responsible for that, not only sodium, other transport mechanism, how it is happening, we must know. As we have discussed that there is a various active processes along the cell membrane, and which transports the solutes, and mostly they are active process and requires protein, carrier protein. In this, regarding that, you should know the tubular transport of maximum. Now what is that? The tubular transport maximum is the maximum rate, at which the solutes are transported through this carrier protein. That is very important to know. And substances which are actively reabsorbed or secreted required carrier protein. And this carrier becomes saturated as the tubular load increases. I'll discuss what is that, how it becomes saturated. And the solutes which are responsible for this transport maximum are mainly glucose, amino acid, phosphate, sulfates. All needs a carrier protein for the transport from the tubular fluid to cell to blood. Now a reabsorption continues. Reabsorption of these solutes from the tubular fluid to the cell, it continues till these carrier proteins are not saturated. Now try to understand. Suppose in a hypothetical situation, I am telling, because for the glucose and the luminal surface of the cell there is 100 carrier proteins which transport the glucose. Now in the tubular load, there is in the tubule there is a 120 solute number. Okay, so one molecule of carrier protein can transport one glucose. So 100 is transported, remaining what will be? There will be 20. So 20 will be remaining in the tubular fluid, which comes out through the urine. So what is happening? If the tubular load is more compared to the carriers, the solutes start appearing in the urine, unable to reabsorb completely. Actually, it is happening in the diabetes mellitus when the tubular load is increased more than the carrier proteins. So carrier proteins are getting saturated, and the glucose, which is not supposed to present in the normal urine, it starts appearing in the urine, and which is called glycosuria. Now the different transport routes through luminal cell. As this is a schematic diagram, we can see the main transport process through this is through the membrane portals or the channels. This is the carrier protein or the channels. These are from the luminal surface to the interstitium it is going, so it is called the transcellular transport. A maximum solutes are transported through that, and few are there which are called the paracellular pathway. And what is that? Through the tight junction. Though it is called tight junction, there are some leaky channels present in this tight junction. And after that tight junction, there is a cellular space which is called the paracellular space between two cells. That's why it is called the paracellular space. When the transport of the solutes and the water occurs between two cells and through this tight junction, this is called the paracellular transport. And this considerable amount of calcium and potassium and also reabsorbed. Along with the water, the aquaporine channels are present. Water is also transported through the paracellular cell along the sodium. So these are main two transport routes. One is the transcellular through the cell. Between two cells is a paracellular route. Next is cellular distribution along the lumen. How it looks. Let's take a look. This is the schematic diagram, and the cells which are present, these are the lumen, and the cells which start with the proximal convoluted tubule. Each nephron has segment is made up of different types of cell which are uniquely perform specific transport mechanism. But you see there are difference in this cellular pattern in the different parts of the tubule, and how and what solutes it transports. According to that, it is differentiated. Cells are differentiated. Now coming to the proximal tubular cell, you can see there is extensive amplification of this cell membrane towards the luminal side, and they are called cilia, and over this cilia there are small microvilli are present. Okay, and the basal side is also invaginated. Within this, their mitochondria are present over there. This ciliated part is called the brush borders. Now coming to this, these are also ciliated and invaginated. This thick ascending limb of Henle. Compared to descending limb, there is less ciliated and invagination, and mitochondria are almost not present. So mitochondria are mainly present where there is a proximal convoluted tubule, and to the thick ascending limb and the distal convoluted tubule. Okay, why? What is the reason that mitochondria are more in this type of tubular cell? And compared to the collecting duct, the less number of mitochondria and less number of cilia. Now particularly we are very much concerned about the PCT cell, or the proximal convoluted cell. The continuation of the last diagram, you see this is a diagram, and this is the diagram of PCT. This is the luminal cell, this is luminal surface, this is a lumen actually, this is a basal border, this is a peritubular capillary cell, more structured one, and this is the called brush borders. These are invaginated. Now you see the huge number of the one nucleus and huge number of the mitochondria are present. Why? Because the mitochondria is the main source of energy, that is ATP, and most of this carrier proteins are required this ATP dependent. Those are ATP dependent. So because of that, the mitochondria's number are more also, because this cell is actively transporting the solute from the lumen to the blood. That's why the new mitochondria are much in number in case of proximal convoluted tubule, in the thick part of the ascending limb, and also the distal convoluted tubule. This is the same continuation of the last slide. In some other direction, this is a brush borders, this is a tubular lumen, this is the epithelial cell, and a huge number of the mitochondria and one nucleus, and the basal side is also invaginated. Before going to start the main transport process, this is a whole idea of from which part of the tubule the sodium is reabsorbed, how much percentage. Actually, this is a schematic diagram of this. As we have discussed, the filtered load of the sodium is 25,500 millimole per day, and the 60% of this, 67% of this tubular load is reabsorbed from the proximal tubule. Okay, and this thin part of descending limb is completely impermeable to the solute but permeable to water. So ultimately when there it is reaching here, then this 25%, this part is not reabsorbed anything. The thick part of this thick ascending limb of Henle, 25% of the filtered load is reabsorbed. After some time, when it is reaching to the distal convoluted tubule, 5% of the sodium is reabsorbed from the distal convoluted tubule, and the remaining 3% is reabsorbed by the collecting duct. So ultimately urinary excretion is 100 millimole, approximately 100 millimole per day, or the 0.4% of the filtered load is remaining in the urine. Completely it is, rest of the part is reabsorbed. Now we are actually going to start about the transport processes. As we have discussed, the sodium potassium pump, presence of the sodium potassium pump at the basal surface of the tubular epithelial cell, how it is situated. Now this is again a schematic diagram. This is the epithelial cell, this is the basal surface of the cells, this is peritubular capillary, and this is the tubular lumen. You see here the concentration of the solutes are more. The after filtration, maximum number of the filtered solutes are present. Still it has not started. Hypothetical situation. I'm telling you, this is the ATPase pump, sodium potassium ATPase pump continuously, which is pumping the 3 sodium outside the cell against a concentration gradient and 2 potassium coming inside. And this potassium, again, leaky channels are present in the both side of the cell, which is maintaining the potassium balance inside and outside the cell. Because of the presence of this potassium ATPase pump, what is happening? Two things is happening. One is this concentration of the sodium inside the cell is only 35 milliequivalent per liter, whereas the outside, because of the filtered load, it is there's 140 milliequivalent per liter. So you can say it is creating the electrical and the concentration gradient. Electrical how? Because of in the resting condition, we know all this inside the cell membrane it is negative because of the protein inside and the continuous potassium going outside the cell, so it is creating negativity. So electrical gradient is also there for this ions, and it is chemically is also very less in concentration inside, so it is creating a chemical gradient. So utilizing this electrochemical gradient, these solutes are going to reabsorbed from the lumen to the cell and going towards the interstitial space and then taken up by the peritubular capillary. And now see, in the peritubular capillary, because of this after filtration, it is almost protein free. Most of the protein are also present in the peritubular capillaries compared to this. So osmotically this is very much high. So what will happen? The water is also reabsorbed very easily by the process of osmosis from this to the peritubular capillary. Now the solute and the processes which are present in proximal convoluted tubule. Mainly this is a schematic diagram of early part of proximal tubule. This is a luminal cell, continuation of the last slide. This is a sodium potassium ATPase pump which is creating a electrochemical gradient inside the cell. So this is a primary active transport. Utilizing this primary active transport, so many secondary active transport are present in the luminal surface. The first one we will discuss about the sodium glucose transporter. This is a secondary active transport or the symport, which transport one molecule of sodium along with the one molecule of glucose. After coming to this inside the cell, the glucose is transported or reabsorbed towards the interstitial space or the blood by this facilitated diffusion which is present at the basal border, that is called GLUT. The next one, the transport ions are the amino acid, the phosphate, lactates, both are through the symports along with the sodium. Now another thing which is not coming inside, which is going outside, actually it is secreted, that is called the H plus ion. Now where the H plus ion is coming, that is very important to know. By the you know, this is the carbonic acid in the presence of the carbonic anhydrase which is present inside this proximal convoluted tubular cell. This is divided into H plus ion and the bicarbonate. This is the this hydrogen ion goes out of the cell by this exchanger. This is exchanger which is going out of the cell, contrary to one molecule of the sodium is coming inside. Another thing you should say, there's the aquaporin. These are called the aquaporin or the water channel. Water is transported by the process of osmosis. Because so much of solute is already present here, it causes the water to drag inside. It's called the solvent drag. Other than this, this is the transcellular pathway, paracellular pathway, water and also sodium. Along with the sodium, water is also transported or reabsorbed through this paracellular pathway. Continuation of the last slide. We can see this is the luminal site. The hydrogen ion is secreted by exchanger, sodium hydrogen exchanger. What the name of this is NHE3, and bicarbonate ion which is synthesized by this process. This is again reabsorbed by with the one molecule of sodium, that is called NCB1. So these transporters are very important to know, and there are so many transporters are there which is transporting along with the sodium, glucose, amino acid and phosphate. So there is one disease actually called Fanconi syndrome. This is a generalized defect in the proximal reabsorption involving this amino acid and the phosphate bicarbonate. So what happened because of this genetic defect, the protein which is helping of this reabsorption, those are knocked out actually. So what happened, thus impaired ability of the proximal convoluted tubule to reabsorb organic and inorganic compounds results in the glycosuria with a normal plasma glucose and hypophosphatemia, hypouricemia, because it is not reabsorbed, so it will be coming through the urine, and the name of this disease is called Fanconi syndrome. Picture of the solute transport is quite different from the early part of the proximal tubule to the late part of the proximal tubule. The chloride which has not been reabsorbed from the early part, mainly it is reabsorbed from the late part of the proximal tubule, and both the transcellular and the paracellular pathways are responsible for that. In the paracellular pathway, it is reabsorbed along with sodium and directly through the tight junction. But here through the transcellular pathway, there are two antipode which is present at the luminal border. One is sodium and hydrogen antipode, another one is a hydrogen chloride antipode. So this again, the hydrogen is synthesized by this process with the presence of the carbonic anhydrase, and after coming inside this chloride, and what is happening, this chloride and potassium, whatever potassium is coming inside, though there is a potassium leaky channel, this potassium again going towards the interstitial space by this another symport called the chloride and potassium symport. Now the thick ascending limb of Henle. We have not discussed about the descending limb, and descending limb there is only, we have discussed already, there's a water only reabsorb, no solutes are reabsorbing, and now we have reached to thick ascending limb of Henle. Now this is very interesting happening here. There is a transporter called 1 sodium, 1 potassium and 2 chloride. That means the 1 molecule of sodium, 1 molecule of potassium and 2 molecule of chloride together by a symport, there is a secondary active transport, is reabsorbed inside the cell. And obviously there is another antipode of this hydrogen and sodium antipode is present. Now what is happening after this potassium and the chloride? Obviously this active transport is primary active is present here. There is a leaky channel and there is a chloride, whatever the two chlorides are coming inside, this is going by the diffusion process. This is a chloride channel. The name of the chloride channel I am going to tell that later. And this chloride recycling is also there by this bicarbonate which is producing by this is taken up by the blood, and then it is the chloride, it is coming inside the cell. And you see here there is a paracellular, through the paracellular pathway there is magnesium and calcium along with the sodium, it is taking up or the reabsorbed through this paracellular pathway. Continuation of the last slide. And this is the thick ascending limb of Henle. We can see this 1 sodium, 1 potassium and 2 chloride. The name of this is NKCC2. Whereas this potassium, whatever is coming inside, it is checking the balance by this another channel, the leaky channel, which is the name of this channel is renal outer medullary potassium channel, ROMK channel. And the chloride which is diffused out of the cell, this channel has also name. This actually of this diffusion process helps by this chloride KB and K1 channel. And the as we told, the calcium and the magnesium is also reabsorbed, and the protein which is present is called claudin. Now there is a disease called Berne syndrome. This is actually a very rare disease. and this occurred due to mutation of the gene for Bartin, and which is an integral part of the protein essential for this chloride KB channel. It's a gene responsible for this protein, and this mutation of this gene causes also a problem in this NKCC channel. So what happened, there is chronic hyponatremia because sodium is not reabsorbed, okay, and this there is a sodium is not reabsorbed here. 25% of the sodium what is reabsorbed from this thick ascending limb of Henle is not reabsorbed here, so there is a hyponatremia and hypovolemia. So because of this there is a activation of the renin-angiotensin system, and ultimately there is resultant increase in aldosterone secretion and causes hyperkalemia, but hypertension doesn't happen. But surprisingly this gene mutation Bartin, it causes also deafness because of the mutation of the ear cells. There is, as we have discussed already, the sodium potassium pump actually creating a gradient, and there is very important the ROMK channel, which is actually needed for the recycling of this potassium, and CI chloride channel Ka and Kb also, it is present in the luminal side, which actually passively transport the chloride to the interstitial space. Now we have reached to distal convoluted tubule. We can see this sodium potassium pump is always present in the basal surface, and here it is also actively transporting the sodium along with the chloride. Mainly the sodium and chloride is imported from this lumen to this cell, and the potassium is recycled to this leaky channel, and the chloride is also taken by this chloride channel, which is present in the basal surface. And surprisingly this this thick ascending limb of Henle and the distal convoluted tubule is completely impermeable to water. Now we have reached to collecting duct. Actually collecting duct has three type of cells. One is intercalated A cell, and there is principal cells, and there is intercalated B cells. And intercalated A cell is mainly responsible for the acid secretion because of the carbonic anhydrous which is present in this cell is responsible for this antipode which is present in the hydrogen potassium antipode. The acid secretion which happens in this hydrogen potassium ATPase pump, which is present in this gastric cell, the same thing, it is present here also, and this is called the part where the acid is secreted, and this is the bicarbonate is exchanged through this chloride, and it is actually taking part of this anion exchanger, and it maintains a negativity inside the cell. And there is also the import which is name is KCC4. The principal cell where this actually ADH, the anti-diuretic hormone acts, and also the aldosterone acts over the cell, and these cell are responsible for the sodium reabsorption with the help of aldosterone, and water reabsorption with the help of ADH. This principal cell is responsible for that, and the aldosterone produces epithelial sodium channel. Explanation of this effect of ADH and aldosterone I have discussed in the later part of my another YouTube link. Please check it, that I have given the link in the next slide. And another cell is called intercalated B cell. Intercalated B cell is also, there is a bicarbonate is exchanged again for this chloride, and this channel is another name is called the Pendrive. So actually this bicarbonate is secreted to make the balance of this pH of the urine. Here hydrogen ion is secreted, and here bicarbonate is secreted, so it's maintaining the pH of the luminal fluid, and this is called the Pendrive. As we discussed, there is a three types of cells, mainly intercalated A, principal and intercalated B cell. And the arrangement favors the secretion of the acid by the intercalated A cell. The hydrogen potassium ATPase pump at the luminal surface, which causes the potassium converts the potassium in situation of the potassium depletion. Intercalated B is completely for this bicarbonate transporter, which creates a bicarbonate and called Pendrive channel. Okay, and epithelial sodium channel, which is amiloride sensitive epithelium sodium channel. Actually, it is present in the principal cells, which is in the presence of the aldosterone. It helps the sodium reabsorption. And now the principal cell, which has two receptor. Principal cell of the cortical collecting duct has two receptors for two hormone. One is ADH and aldosterone, and with the help of that ADH, it reabsorbs the water, and with the help of the aldosterone, it reabsorbs through the epithelial sodium channel. It reabsorbs the sodium. So the detailed discussion of this too is done in my another link, YouTube link. Please check it for that. And the aldosterone escape is also discussed in this link. We have now we are discussing about the water reabsorption by the tubule, mainly by the process of osmosis. The movement of the solvent molecule from a higher concentration to the lower concentration of the solvent molecule, where the solute concentration is more. How the solute concentration is more and where it is more? After reabsorption of this so many solutes from the tubular lumen to the interstitial space, solute concentration is becoming more, and because of that, automatically this water will be reabsorbed by the solvent drag by the process of the osmosis. There are two types of osmosis process of this water, where it is happening. One is water is obligatory water transportation or reabsorption. Water is obligate to follow the solute, where the solute concentration is more, and that is the first part of this PCT. It is happening. Water is reabsorbed along the solutes, and another one is 90% of this water reabsorbed by this process and occurs in the PCT, descending limb of Henle. Descending limb of Henle is permeable to water compared to the ascending limb. There is ascending limb thin and thick, but both are impermeable to water. Put it in your mind. And another way of transport is facultative water reabsorption. And what is that? The transporter or the hormones, which is called the faculty. That's what name has been given. The facultative reabsorption is required, and it is remaining 10% of the water is reabsorbed by this facultative water reabsorption. It is happening in the collecting duct. It is along this ADH is a hormone which is responsible for the water reabsorption, and also with the water, the sodium water is also reabsorbed through this. So that's why it is called the facultative water reabsorption. And here we need this aquaporin. So aquaporin 1 is also required in this obligatory water transport, which is present in the PCT. There is aquaporin 1 and another aquaporin which is present in this part of this aquaporin 2. This is a schematic diagram. You can see here this is a glomerulus at the proximal convoluted tubule. Because of the solvent drag, this water is reabsorbed along with the other solutes, and this creating osmolality, increase in osmolality in the interstitial space and the blood. So water is reabsorbed, and that is a obligatory water osmosis. Here also, because of this medullary interstitium is also solute concentration is more, so it is going out of the tubule to the interstitial space by this obligatory water reabsorption. And you see here the ascending limb, both the thin and thick, these are impermeable to the water. And here coming to this distal tubule, because of the sodium, again it is reabsorbed. Sodium and the chloride is reabsorbed because of the solute concentration. Again it is causes the solvent drag, but very little of this water is reabsorbed here in the distal portion. And this is the facultative reabsorption of the water with the help of the ADH, and here it is by the aquaporin 2, here it is by the aquaporin 1. This part is by the aquaporin 1 in the presence of the aquaporin 2 in the collecting duct. Now we are going to discuss about the very interesting topic, what is called diuretics. Diuretics, that by the definition, it is any substance which increases the volume of the urine. It called diuretics, or otherwise you can tell it is a water pills. Now the types of diuretics. In this diagram you see the 1, 2, 3, 4, 5 types, and where they are present and how they are. Actually the diuretics block some enzyme or block some channels which is responsible for the reabsorption of different solutes. So this causes the increase in urine volume, and that's why it helps whenever the volume of the body blood volume increases, that diuretics is prescribed to decrease the volume by increasing the urine volume. And how it acts by blocking of this channels which is responsible for the reabsorption of different solutes. So first we can see the first is acetazolamide, where it acts, it blocks. Acetazolamide is an inhibitor of the carbonic anhydrase, which is responsible for this hydrogen ion production inside the PCT, which blocks this. And instead of there is a antipode is present in the PCT, which secretes one ion of hydrogen ion instead of one ion of sodium. So if this carbonic anhydrase is inhibited, the sodium which is reabsorbed against the hydrogen ion, which won't be there, so it is a production of this urine volume will increase because of this. And this second type is osmotic agent, that is a mannitol. Because of this obligatory reabsorption is happening here, so what will happen, the mannitol, if it is given, mannitol is completely filtered and not reabsorbed through this. So the presence of the mannitol, which is osmotically active particles, which drags the water, it blocks the reabsorption of the solvent drag of the water from the tubule to this interstitial space. So water mainly remains in the tubule, rather reabsorbed from the tubule. So the second type is osmotic agent, mannitol. The third type is loop agent, or the loop diuretics called, because it works in the thick ascending limb of Henle loop, and which it blocks the 1 sodium 1 potassium 2 chloride transporters, which helps the dragging of the water with this solute transporter. So it blocks this. This is called the loop diuretics. Another one is, a fourth one is thiazide, which blocks the sodium and chloride transporter, which is present in the distal convoluted tubules. And there is an antagonist of the aldosterone, and here collecting duct, and antagonist of the ADH also present. Now better classification of this, where it is acting, you can check it here. PCT, as I say, the carbonic anhydrase inhibitor, actually it is present over, which is secreting the hydrogen ion in the PCT. It blocks it, and this is called acetazolamide. Thick ascending loop of Henle, loop diuretics is furosemide. And thiazide, this is a thiazide, hydrochlorothiazide. And actually the side effect of this is, actually this too much of potassium is secreted through the urine, so there is a hypokalemia. It means the side effect of this to diuretics. But here in the potassium cortical collecting duct, which is a potassium sparing diuretics it's called, it doesn't cause the hypokalemia. This name of this diuretics is spironolactone. Osmotic diuretics, it acts over the PCT, late part of the PCT, whereas the solvent drag, it stops the solvent drag in the late part of the PCT and also in the descending loop of Henle. And other two diuretics, which is acting on the collecting duct, and this is antagonist of the ADH and antagonist of the aldosterone. At the very beginning, I have discussed that the sodium is a main solute which maintains the extracellular fluid volume, rather the blood volume. So the main regulator of the solute concentration, or the regulator of the blood volume, is one of them is renin-angiotensin system. And the very short, how the renin-angiotensin system works: when there is a renal blood flow is decreased, or the renal sodium concentration is decreased, and the juxtaglomerular apparatus senses that, and it starts secreting renin from the juxtaglomerular cell. This renin acts on the cell, which actually converts the angiotensinogen to angiotensinogen 1, and this angiotensinogen 1 enzyme, when this blood full of angiotensinogen 1 is passing through the lungs, it converts the renin in the presence of the angiotensin converting enzyme. Is when it is passing through this angiotensin 1 is converted to angiotensinogen 2. This is angiotensinogen 2 is a very powerful vasoconstrictor, okay, and this is converted to this angiotensin 3, and this angiotensin 2, this acts on the renal cortex, and where adrenal cortex, which acts on the adrenal cortex and secretes the aldosterone. And as we know, aldosterone acts on the principal cell of the collecting duct, and it produces the epithelial sodium channel, and which is responsible for the sodium reabsorption for the collecting duct. And that way, whenever the sodium concentration is less, by inhibiting this production of this aldosterone increases the sodium reabsorption from the collecting duct, and the sodium concentration is restored. Another regulatory mechanism for the solid concentration of the body or the blood volume is tubulo-glomerular feedback. Here we can see the diagram. This is the glomerulus, and this is a Bowman's capsule, and this is the part where it is a distal convoluted tubule. Actually, this is the part of the distal convoluted tubule which is nearby this the macula densa cell. So macula densa cell, the glomerular juxtaglomerular cell, and the Lassus cell consist of the juxtaglomerular apparatus. That is very important. Now, what is happening if the renal blood flow is increased? Okay, so what happened? This increases the renal hydrostatic pressure also in the afferent arterioles, which increases the GFR. If increases the GFR, obviously the solute concentration of this ultrafiltrate also increases, and this will sensed by the macula densa cell. The concentration of this, actually the sodium this macula densa cell acts as a chemical sensor which is present in the distal convoluted cell. The macula densa cell senses the salt release in the vasoconstrictor chemicals, and afferent arterioles constrict, and after afferent arteriole constrict, and what is happening? Decrease in renal blood flow, decrease in hydrostatic pressure, and decrease in GFR. Now, what is the mechanism of tubulo-glomerular feedback? What is happening? Arterial pressure has increased, so GFR has been increased, and because of the GFR, the tubular load of the sodium chloride and potassium has been decreased, and which has been transported to the distal convoluted tubule. There also it has increased. So this is sensed by the macula densa cell, which is present in this distal convoluted tubule, and this there is a transporter. This is a NKCC2, which is mainly transporting the sodium chloride and potassium to inside the cell, and this transporter, secondary active transport, actually utilizing this primary active transport. And because of the too much of solutes are reabsorbing, this too much of breakdown of ATP, and this too much of breakdown of ATP causes breaking down the ATP for the adenine, and adenine receptors are present on the Lassus cell, which is present on this juxtaglomerular apparatus. When breaking down of the ATP, this adenine from the macula densa cell, it is diffused to the adenine receptor, A1 receptor, which is present on the glomerular cells, the Lassus cell. It is sensed by that, and ultimately it is secreting this too much of calcium from this to the smooth muscle of the afferent arteriole. Because of this voltage gated, so the calcium channels are opened, and the calcium is going inside to the smooth muscle. Ultimately, increase in calcium concentration causes smooth muscle contraction. That is smooth muscle contraction of afferent arteriole, and because of the afferent arterial contraction, what is happening? Ultimately, GFR is decreasing. So signal is the increase in solute concentration in distal convoluted tubule. Transmission is through the macula densa cell. Mediator is A1 or adenine. Adenine receptor are present in the Lassus cell, which is a chemical sensor, and ultimately there is a vascular smooth muscle, increase in the calcium concentration of the vascular smooth muscle of afferent arteriole, causing constriction of the arterial afferent arteriole and decreases the GFR. Two terms you should know in between. One is a pressure diuresis. Pressure diuresis is nothing but the increase in the urine volume by the water volume or the blood volume if it is increasing, which is increasing ultimately the blood pressure, and blood pressure by the process of this two regulating mechanism. The urine volume is also increasing, and this is called the pressure diuresis. Another term is called the pressure natriuresis because of the sodium. I told before that sodium is the most osmotically active solutes. Because of the sodium, if it is coming through the urine, it drags the water and it maybe the stops the water reabsorption from the tubule to the blood, and this causes the pressure natriuresis, increase in pressure, which is maybe due to the sodium concentration, and ultimately by this two regulatory mechanism the concentration of the sodium is also increasing in the tubule and increase in the pressure, and ultimately increase in the sodium excretion. Ultimately this too decreases the volume and decreases the pressure. Now the factors which regulate the sodium excretion, first and foremost is the hormonal factors. Which hormone is mainly responsible for this sodium excretion is the aldosterone. Aldosterone is secreted from the adrenal cortex, and from the adrenal cortex few amount of the glucocorticoid, because this is secreting from the adrenal cortex, it has also action like the mineralocorticoid or the aldosterone. And the sex hormone, again it has a few percentage of this sex hormone which is secreting from the adrenal cortex, has aldosterone like action. And other hormone is angiotensin 2, which mainly acts on the stimulates the adrenal cortex for secretion of the aldosterone, and ultimately it causes the sodium reabsorption, which is aldosterone is a sodium retaining hormone. And the angiotensin 2 also is a potent vasoconstrictor. So because of the vasoconstriction, GFR decreases and the sodium excretion, the reabsorption is regulated, and it also stimulates the sodium potassium ATPase pump, which helps to maintain the electrochemical gradient. And another one is ANP, atrial natriuretic peptide, which decreases the sodium reabsorption by inhibiting the epithelial sodium channel at the collecting duct, which I have discussed elaborately in my another YouTube link. Already I have shared that. And sympathetic stimulation, which the beta fiber, which increase the renin secretion, and also sympathetic stimulation causes the constriction of the afferent arteriole by decreasing this GFR. And diuretics, already we have discussed the function of the diuretics, which increases the urine volume. In this diagram you can see that the name of the different diseases, and it is the protein which is responsible for the transport of different solutes which are present in the different parts of the tubule, and if it is the gene is knocked out, then the disease it happens like this: the Fanconi syndrome, Bartter syndrome, and others also. When it is distal convoluted tubule, the sodium and the chloride transporter, if it is knocked out, and then it happens these diseases. Now ultimately we have reached to the end part of this discussion. Now it is time to summarize. This is the schematic diagram. Proximal tubule, the whole filtered whatever it is there, the glucose is reabsorbed 100%, amino acid 100%, urea 50% is reabsorbed from this proximal convoluted tubule. Sodium 67 to 70% is reabsorbed from the proximal convoluted and the late part of this proximal straight tubule. And the phosphate, which is phosphate and the chloride, it has been reabsorbed at the late part of this proximal tubule. And this is water. 70% of this water is reabsorbed from this proximal convoluted tubule. Descending limb is permeable to the water, not to the solute. You have to remember that. Ascending limb, both thin and thick, are impermeable to water. The solute which is reabsorbed from this thick ascending limb is 25%. Yes, 25% of the sodium, rest potassium, and the calcium and magnesium is reabsorbed from the thick ascending limb. 5% is from the distal convoluted tube of sodium is reabsorbed, and 3% of this is sodium is reabsorbed from the collecting duct. And the secreted part is one is the hydrogen ion, the potassium, and the drugs, which is secreted mainly in the distal convoluted late part of the distal convoluted tubule and the collecting duct. So here we end the kidney, how it handles the solute by the reabsorption process and the secretion.